Hello everyone. Let's talk about part 2 of lecture 3 lymphatic system, innate and adaptive body response. Immune system uh, has two intertwined intrinsic system, innate and adaptive defense system. They release and recognize defense molecules and proteins that alert cells of the adaptive system to foreign molecules. Innate defense system has two lines of defense. First, external body membranes such as skin and mucosa, and second is antimicrobial proteins, phagocytes, and other cells, and they invade spread of invaders. And inflammation is the most important mechanism. And adaptive defense system is the third line of defense, attack particular foreign substances, and it takes longer to react than innate system. And humoral body immunity concerns with the B cells making plasma cells to make antibodies, and cellular immunity concerns T cells, uh, which directly kills the cancer cells and virus infected cells. Let's talk a little bit about uh, surface barriers such as skin and mucous membrane and secretions, and those are the barriers for microbacteria to spread. And like protective chemicals, they inhibit and destroy microorganisms such as skin is a little acidic and has a secretion, and enzymes in the mucous membranes which will kill the bacteria, and also uh, like for instance mucosa in respiratory system, they have hairs and of the cilia and the upper respiratory tract especially, and they have a mucus containing a lot of barriers for bacteria to enter a deep structure of the lung. And innate internal defense, those cells and chemicals are like natural killer cells and antimicrobial proteins such as interferons, etc., and fever and inflammatory response. Those are all very important in terms of deterring the bacteria to enter first and second line of defense. Phagocytes are neutrophils. They are most abundant, but macrophages are uh, basically the major ones which gives phagocytic action, and they are derived from monocytes. There are two types: free macrophages, which wanders through tissue space, and especially like alveoli macrophages. Some of them are fixed macrophages, and they are permanent residents of some organs such as liver, Kupffer cells, microglia in brain. So mechanisms of phagocytosis involved phagocytes adhere to pathogen and then engulf by through the pseudopod and forming a phagosome inside of the cell, and then it fuses with a lysosome, uh, creating a phagolysosome, and then enzymes, especially hydraulic enzymes of the lysosome, now kills those bacteria within the structure and creating a residual body, and so that cells can now exocytose those residual bodies. Helper T cells cause release of those enzymes, especially in the case such as producing the oxygen radicals or by increasing the pH. Natural killer cells are non-phagocytic cells. They attack cancer cells and virus cells to die, so-called apoptosis. And they also secrete the chemicals to enhance inflammatory response. And those inflammatory responses are triggered when body tissues are injured, and then body wants to prevent the spread of this damaging agent, and also make sure that you clean up those cell debris and pathogens, and then alert the adaptive immune system to set for next stage to repair. However, the signs of inflammation creates the redness and heat and swelling and pain, and normally they begin with the chemicals released by extracellular fluid by injured tissue and immune cells and blood proteins. And inflammatory mediators are those like prostaglandins and complement proteins, and they normally act on dilation of arterioles and creating the redness and heat of those inflamed area and making the capillaries leaky, and so that white blood cell can come to that area, and therefore that the fluid permeability increase, causing the edema and so swelling, and then it pushes the nerve endings so that one feels the pain sensation. And also next is that chemicals will now deliver those chlorine proteins and complements to actually clean up the area so that repair can occur and so bacteria cannot spread. Phagocytic mobilization involves first neutrophils come first and then macrophages come next. Steps for phagocytic mobilization involves first leukocytosis, that is the release of neutrophils from bone marrow in response to signals from injured cells, and second stage is the margination, which involves the neutrophils cling to the walls of capillaries in inflamed area, and third is diaphysis of neutrophils, and that's the neutrophils squeeze out of the capillaries, and then chemotaxis, which neutrophils will sort of follow through chemical attraction so that it promotes the chemicals to go to where actually damage occurred. And basically inflammation is basically activated the complement system, and then adaptive immune system will follow. Antimicrobial proteins are interferons and complement proteins, and they attack microorganisms directly and kill them. Interferons are a family of immune modulating proteins and have different kind of functions. And normally virus infected cells secretes the interferon to warn the neighboring cells there is a virus, and then what happens is that interferon will now enter to the neighboring cells and then produce the proteins which block the viral production and degrade the RNA for those viral RNA. And so, and also interferons will activate the natural killer cells, and also activate the macrophages, and since interferons activate the natural killer cells, and macrophages indirectly fight the cancer. So therefore that interferon can artificially made interferons can use to treat the hepatitis C and like multiple sclerosis or types of also cancer. Complement system are made of 20 different proteins that circulate in inactive form. They are, as you see, consist of many proteins here, and basically fever is abnormally high body temperature, and it is a systemic response to invading microorganisms due to the pyrogens secreted by bacteria. And basically complement system generate the inflammatory chemicals that amplify all aspects of inflammatory response and kill bacteria and certain type of cells by cell lysis. and also calls for innate and adaptive immune response. And there are three pathways to activation. First is classical pathways, uh, involves the antibodies binds to invading organisms and to complementary components, and they are called complement fixation, and that's the first step of activation. In lectin pathways, basically lectins are produced by innate system to recognize the foreign invaders, and basically uh, they interact with activated proteins such as C3, B, D, P, interact at the surface of the microorganisms. And the other is the alternative pathways, which doesn't have those uh, lectin pathways. However, together, um, each pathway converge on C3, which is the common pathway, and then they divide into either C3A or C3B, and then common terminal pathway is initiated by enhance the inflammation by inducing phagocytosis and causes a cell death. And that is occurred by inserting membrane attack complex called MAC, okay, of those into the membrane of the bacteria, so that there's a hole in the membrane, so that influx of water occurs that causing the cell to lyse. So this is the basically how complementation works. Antigens are substances that can mobilize adaptive response and provoke immune response. Complete antigen have immunogenicity normally, or reactivity. Immunogenicity means the ability to stimulate the specific lymphocyte to produce antibody, and reactivity means that basically to react with antigen and antibody to create the immune reaction. So those examples can be foreign proteins, sugars, lipid, and nucleic acid. Another term is a hapten, which is incomplete antigen. Haptens are small molecules. They are not immunogenic by themselves. However, when they attach to the body proteins, and combination can create some harmful attack to immune system. Example can be poison ivy, animal dander, detergent, and cosmetics. Only part of that antigen can be act as antigenic determinants to be immunogenic. So basically they act like antibodies, and the lymphocytes are receptors to bind them as like enzyme binds to the substrate. Most occurring antigens have a numerous antigenic determinants that mobilize several different lymphocyte population and form different kinds of antibodies against them. And then normally that large chemically simple molecules like a plastic has a very low immunogenicity. So normally people can insert the plastic bag in their body parts and then try to make it look good, but it doesn't have any rejection. Lymphocytes normally originated from B cells and T cells as a precursor in red bone marrow, and they migrate either into thymus or some stayed in red bone marrow, and then B cells develop from red bone marrow and T cells mature in thymus. And then secondary lymphoid organs normally sees the some antigen, and then antigen will trigger those lymphocytes to be activated and in a lymph nodule, and then proliferation of that particular B cells will now become plasma cells to produce many millions of antibodies, so that it can attack that antigen of, at that point. The maturation of T cells in thymus has to go through two types of selection before they can confer cellular immunity. First, they have to go through positive selection. That means that T cells must recognize their uh, self major histocompatibility complex called MHC. So if they do not recognize their self MHC as a self, then they have to die. So that's called positive selection. And also negative selection involved with that if they recognize their self antigen, then they have to die. Otherwise, they will cause the autoimmune disease because you're not supposed to attack oneself. You're supposed to attack something else. So this is called negative selection. B cells mature in red bone marrow and they are positively selected if successfully make antigen receptors. Those that are self-reactive has to be eliminated by cell death. And so those immunocompetent B cells which has not exposed to antigen are called naive, and also they normally exist in secondary lymphoid organs like lymph node and spleen, so that they can increase the chance to meet those foreign antigen if they are in the system. And then first, when those naive lymphocytes first encounter with this foreign antigen, they are selected for further development. Those which fits with that particular antigen will be selected, so that is called activated B cells, and they become plasma cells. Those plasma cells will now secrete the huge amount of antibodies, which now will actually eliminate those antigen which naive B cells encounter at the beginning. And also part of those clones that do not become plasma cells become memory cells, and they provide immunological memory, and then they can actually wait for the future exposures to the same antigen. Immunological memory is very important for the time that how antibody can actually react to the antigen exposure. So primary immune response involves normally cell proliferation and differentiation upon first antigen exposure. And those lag period, as you see here, takes about 3 to 6 days, and peak levels of plasma antibodies are normally shown about in 10 days. And then antibody levels then decline. And secondary immune response involves re-exposure to same antigen. It gives a lot faster, more prolonged, and more effective response. Sensitized memory cells respond within an hour, and the antibody levels peak in 2 to 3 days at much higher levels, and antibodies binds with a great affinity and remain high for weeks to months. Active humoral immunity involves when B cells encounter antigens and produce specific antibodies against them. There are two types of active humoral immunity, either naturally occurring response to bacteria or viral infection, or artificially acquired response to vaccines of dead or attenuated pathogens. Active humoral immunity involves this dead or attenuated pathogens, and passive humoral immunity are normally ready-made antibodies introduced into body, and protection ends when antibodies degrade. Antibodies are normally showing Y-shaped structures. They are normally 4 peptides linked by disulfide bond, consist of 2 heavy chains and 2 light chains. and also variable area at end of each arm combined to form two identical antigen binding sites. And there are five types. um, IgM is the biggest one, pentamer, and is the first antibody released, fixes the complementation. IgA normally exist in mucus, and they can be a monomer and dimer, which prevents the entry of the pathogen. IgD normally attached to the B cell surface as a receptor. IgG is the most abundant antibody. 75 to 85% of the circulating antibodies are IgG, and they cross the placental barrier. IgE is the monomer which generate the allergies and parasitic infections, and these are the ones which cause the basophils for histamine release. Uh, Antibody binds to antigen and forming an antigen antibody complex, and then they will inactivate the um, antigen by agglutination, making little globules, or they can neutralize by covering the surface, either bacteria or viruses, or they can form a lattice structure to make it heavy to get out of the solution as a precipitation. So all these things will enhance phagocytosis and inflammation process, or they can actually enhance the complementation fixation, forming the membrane attack complex, so the cell can be dead. Um, T cells, when it mature in a thymus, and they become two population. One is uh, CD4 T cells, and the other is CD8 T cells. CD4 cells will interact only with uh, antigen presenting cells containing class 2 MH2 major histocomplex MHC proteins, so that CD4 can only bind to class 2 MHC. So this is how it can actually work as a helper CD cells, which is important for the linking uh, humoral and also cellular induced immunity. And CD8 cells will interact with only class 1 MHC antigen presenting cells, especially those can be dendritic cells here. And guess what? CD8 and MH1 class will actually create the cytotoxic T cells, so that direct cell killing can occur. So this is very specific, and so that you have to understand this interaction. This shows you a table. As you see that MHC class 1 uh, is only present all nucleated cells, so that only red blood cells do not have this, but all your cells in your body has MH1 class saying that's you. Okay, and class 2, MHC class 2, is present only antigen presenting cells, such as dendritic cells, macrophages, and B cells. So here's very important. MH1 class can display that antigen to cytotoxic T cells containing CD8. Okay, so this is very important. In the case of um, MH class 2, will actually present those antigen to CD4 cells, which is the helper T cells, and this is how exogenous uh, antigens can be phagocytized. And in the case of CD, in the case of MH class 1, endogenous uh, pathogens such as cancer cells and virus infected cells can be presented to the cytotoxic cells, so that cytotoxic T cells can kill them. So this is the summary of how primary immune response work. When antigen enters your system, it triggers either innate defense mechanism. We talked about surface barriers and internal defense of that involving with the B cells producing the antibodies, or basically deterring the primary defense, like a skin and then mucous membrane, or adaptive response involved with phagocytose with that antigen, and then those antigen presenting cells has to first work through CD4 cells by binding to MHC class 2 to CD4, and then CD4 cells will help for B cells to produce the antibodies. So, and also it will also help helper T cells to actually activate the cytotoxic T cells containing CD8, so that it can activate killing by producing the membrane attack complex.